Well, good morning, everyone. Welcome to our webinar. The topic that we wanted to speak about this morning was how healthy plants create soil. I'm going to be presenting quite briefly and then leave lots of time for question and answers. So, for decades, the idea within biological and organic agriculture that it takes a healthy soil to grow a healthy plant, and the focus should be on building soil health. Let's use compost, let's grow cover crops, let's add manure, let's keep our soil covered all the time so that we develop a really healthy soil. And then that healthy soil will help us to produce healthy crops and to produce healthy plants. This idea isn't entirely incorrect, but it has resulted in an understanding that I believe is very incomplete. And what has been missing from the conversation is the realization that compost and other things create soil health, which then creates plant health, but actually it's healthy plants that create healthy soil. It is the process of photosynthesis where plants are sequestering carbon dioxide from the air, producing sugars, producing carbohydrates, and sending a significant proportion, perhaps even the majority of the total carbohydrates they produce out through the root systems as root exudates to build soil organic matter. This is really what builds soil health. So I think what has been missing is the question about what is really the root cause, what's the root driver. And the point that I want to make is that the engine that drives soil health is plants. It's not soil. In reality, without the contribution of plants, if you wouldn't have plants photosynthesizing and sending these sugars out in the soil system, soil would be nothing more than decomposed rock particles. You'd have sand. And so I think this is a very important piece that is beginning to be better understood, but still hasn't been widely recognized enough. Why is it so important? Because when we understand that it is healthy plants that drive the ecosystem, we will begin focusing on producing exceptionally healthy plants, exceptionally healthy crops, rather than focusing on soil health. Growing really healthy crops is the pathway to developing soil health. It's the engine that drives the system. And if we focus exclusively on building soil health and expect the plant health to be a secondary outcome, we can get to the same place, but it's going to take 10 or 20 years. It's a process of decades rather than a process of 3 or 4 years. And I think this is really one of the key aspects that has allowed us to be so successful in our work at advancing eco-agriculture, and what we've really developed a reputation for is helping growers see rapid responses with disease and insect resistance, achieving higher yields, achieving better plant quality. All of those happen for us in the systems that we put together very quickly. We expect to see results in the first year. We want to see results now, immediately. And then not only do we want to see immediate economic results, but also we expect to see soil organic matter build very quickly. And the idea has been historically that this is a process which takes decades or perhaps even generations to build soil health and to build soil organic matter. Our observation, our experience has not been that is the case, but that in fact we can get results in the first year, the second year, the third year. We can build soil very quickly. And the difference is in that instead of focusing on building soil health, we've instead focused on increasing plant health and letting that contribute to soil health. So how does this work? How does it function? I put together a diagram, I've spoken about it before, called the biological cascade, where we describe what we are seeing in soil and plant ecosystems and how it was really designed to function. So the first step in this process of the biological cascade is where we have plants that are at what we describe as being full capacity photosynthesis. It's improbable that we will ever have plants in an outdoor agricultural environment that is at 100% photosynthetic capacity, because very generally we have limitations, either inadequate carbon dioxide, inadequate water, perhaps inadequate sunlight when we have cloud cover, etc. And then in addition to those three factors, we can often also have inadequate nutrition, not enough manganese, not enough nitrogen, magnesium, sulfur, etc., to facilitate 100% photosynthetic capacity and to enable the plant to perform at that level. So what we're talking about is most plants today are somewhere what we've come to accept as being common and normal is plants that are in the neighborhood of about 15 to 20% photosynthetic capacity. And while we're never going to be at 100%, or it's unlikely, it is possible to go much higher than 15 to 20. We can easily move plants up to 40, 50, 60% photosynthetic capacity. And particularly when we have supportive weather conditions. So when that happens, we get this spike in photosynthesis. And as you can imagine, when you go from 20% to 60% photosynthetic efficiency, that means you get 3 times the volume, 3 times the quantity of photosynthate, of sugar production, in each 24-hour photo period. When that happens, the sugars now move down out through the root system as root exudates in larger and larger concentrations and result in very aggressive bacterial populations and bacterial digestion in the rhizosphere. There's a lot of really interesting math behind this movement of sugars to the root system, but just the simple headline highlights version is we know that in a plant that is at, let's say, 20% photosynthetic efficiency, depending on the stage of plant development, during fruit fill, it can be 0, but during the framing stage, as much as 70% of the plant's total photosynthate production in each 24-hour photo period can move out through the root system as root exudates. That's for a plant that's at 20%. That proportion can move even higher when overall photosynthetic efficiency is greater than, let's say, if it's 60%. So when a plant moves from 20% to 60%, it means you get triple the sugar production in every day. Does that mean you get triple the yield? It's unlikely, probably not. So if you don't get triple the yield, you're also unlikely to get triple the plant biomass. Where does all the additional sugar go? All the additional sugar, you'll get some increase in yield. You'll get some increase in plant biomass. It'll vary depending on which species and crop we're talking about and other factors, of course. You might get a 30% or a 50% or perhaps as low as a 10 or 20% yield increase. So all the additional sugar that isn't kept stored in the leaves is moved out through the roots as root exudates. You can move a large quantity of sugar into the rhizosphere and trigger bacterial digestion. So then this bacterial digestion produces what is termed a mineralization response in the soil. So essentially, when bacteria consume these sugars that are coming out from the root system as root exudates, we know that these sugars are largely carbon, hydrogen, and oxygen. When you consider glucose, that's C6H12O6. And of course, there are many different carbohydrates and enzymes and other compounds being transmitted into the soil as root exudates. But what isn't present in root exudates in any appreciable quantities is minerals. There's very little, bordering on non-existent levels of calcium and manganese and zinc and copper and magnesium, etc. And yet bacteria need these minerals to form their own bodies. So the bacteria, as they consume the carbohydrates, they extract minerals from the soil mineral matrix and incorporate them into their own cells. And then these microbial cells are now incorporated back into root systems and take the nutrients with them. So from this mineralization process, the next step is when plants begin absorbing these microbial metabolites, they now become very energy efficient. They can now access an abundance of nutrition. They're producing with photosynthesis, they're producing more energy than they need to sustain themselves. And they begin storing the surplus energy in the form of lipids, plant fats, and oils. So we can see this in the field visually when we have leaves that produce this very waxy, oily sheen on the leaf surface. They become very resistant to airborne pathogens at this point. But what happens from a soil biology perspective is that now the plants also begin transmitting lipids out through the root system and into the rhizosphere as well, in addition to the carbohydrates and everything else. So when we have lipids moving into the root system, something really interesting happens because bacteria aren't able to digest lipids. So now this will trigger very strong fungal digestion, and we get increasing colonization of mycorrhizal fungi and other types of beneficial fungi in the soil profile that begin consuming the lipids. And fungal digestion is quite different from bacterial digestion in that when fungi ingest something, they metabolize it and digest it and break it down over and over and over again until it reaches a point where it can't easily be broken down any further. It's only at that point that they release it back into the soil profile. And this fungal digestive process is called humification because the fungal digestion of these lipids and along with carbohydrates and other high carbon plant compounds is what results in the formation of stable organic matter. Whether we're referring to glomalin or any of the list of various compounds that aggregates soil particles and produces stable organic matter, these are generally a result of fungal decomposition of healthier plants that have a higher fat content, a higher lipid content. So what this means is that you want to produce really healthy plants that have the highest fat content possible, the highest lipid content possible. It also means that the quantity of organic matter that is developed in a soil from a given crop has nothing to do with the quantity of biomass that is present. It has everything to do with the quantity of lipids that are present. So if you have a field, one field that has a cover crop that contains a total of 2,000 pounds per acre of dry matter, but that 2,000 pounds of dry matter contains 6% fat content, lipid content, or fat content on a dry matter basis, you will form and produce the exact same amount of organic matter as a field that has 6,000 pounds of dry matter at only 2% fat content. So it becomes really clear that if we want to build organic matter quickly, having a higher fat content plant is a very important part of the process to getting there. And this, of course, is at the foundation. The interesting point when you think about this entire cascade, the very first step at the top of the cascade is photosynthesis. When we increase the plant's photosynthesis, then all the rest of this happens on its own, happens automatically. Accelerating photosynthesis is, I think of this as being our tool to hack the system. It's how you can speed up the entire process and make everything happen very quickly. It's becoming increasingly clear and very important to me to realize that when we really focus on plant health, two things happen. One is we get immediate economic results with the crops that we're producing. And secondly, we get our biggest soil response as well. We get a much bigger soil response when we work with plants than we do by adding compost or by adding microbial inoculants. Because this is really, photosynthesis is really the engine. It's the engine that drives the entire system. It's the only process by which we can bring new energy into an ecosystem. So those are the thoughts that I wanted to share. I'm going to switch over to Q&A. I think one of the questions that is often, that has been asked in the past when I've talked about this topic is, okay, this is great. I understand that I need to increase photosynthesis. How do I do that? The headline version of the answer, again, it's a multifaceted answer, as are any real answers regarding agricultural ecosystems. There are four major segments of imbalances that reduce photosynthetic efficiency. The first and the most common is when you have inadequate carbon dioxide. The second is when you have inadequate water. The third is when you have inadequate sunlight. These are all environmental and ecologically related factors that, while there's limitations on what we can do about sunlight, we can in some environments with high value crops. Of course, with water, we can talk about water use efficiency. And actually, I'll speak about water use efficiency for just one moment. The quantity of water that plants actually need is significantly less than the quantity that most crops absorb. The reason for this is because many crops require or use much higher amounts of water because of imbalanced nutrition. So, for example, if a corn plant, or any plant for that matter, absorbs greater than 60% of its total nitrogen requirement, total nitrogen supply, in the form of nitrate, it increases the water requirement by 50%. So just to translate into a hypothetical situation, that means if you have a grapevine that normally would need 10 liters of water per day, you now need 15 liters of water per day. You've just significantly increased the water requirement because of the nitrate that is present in the soil and within the plant, because the plant requires so much additional water to produce energy to convert the nitrate into proteins and amino acids within the plant. So I'm just using nitrate as one example. There are other examples of nutritional balances that have a significant impact on water. The short version is that when nutrition and biology is managed well, most commercial crops could produce the exact same yields of a higher quality plant with about half of the water that they currently use. And when we think about the water situation that we have in much of agriculture, that's a really significant statement and a very important piece to consider. Carbon dioxide, many soils lack adequate active carbon, microbially active carbon, and often aren't able to release enough carbon dioxide for when the plants actually need it. And then, of course, we have tillage and other pieces that happen on some soils where we lose too much carbon dioxide when the plants aren't capable of absorbing it. And then the fourth piece is manganese. I'm separating manganese out from the other minerals because it is so foundational to the photosynthesis process. When plants absorb water from the soil to be used in photosynthesis, the first step is the water molecule needs to get split into H and OH. And this process is called water hydrolysis. Manganese is the enzyme cofactor that is responsible for water hydrolysis. This means even if you have perfect water, perfect levels of carbon dioxide, perfect amounts of sunlight, when the plant has low levels of manganese, manganese immediately becomes the limiting factor for the quantity of sugar that a plant can produce in 24 hours. And then following manganese, we have all of the additional minerals as well, a broad array: magnesium, sulfur, zinc, copper, calcium, etc., all important in some manner to, if not directly in the photosynthesis process, then indirectly to support it. Those that are directly involved would be magnesium and nitrogen and iron. And perhaps in a very strong support role, while not directly involved, but in a very strong support role, would be phosphorus. So the most important nutrients to manage if we're not seeing a strong photosynthesis response that we're looking for is first manganese, the trace mineral manganese, and then magnesium and iron and nitrogen, supported by phosphorus, and then the other minerals ranging in priority below that. And I would suggest that the majority of the crops and soils that we've worked with are not capable of supplying enough iron and manganese to the crop. So You may have a soil analysis that shows you have a generous, even excessive levels of manganese and iron, but you can't use that as an indicator for whether the crop has enough. And you also can't use a tissue analysis as an indicator that the crop has enough iron or manganese. The only indicator that I believe is really accurate is either a sap analysis, which does detect the differences between reduced and oxidized forms of iron and manganese in the plant structure, or you can do a test spot, a test application with iron and manganese and see what happens. There's a question, going to jump to Q&A. Quite a few have come in. Thank you for that. First question from Tuomas. Is there a way to measure lipids in the field, like bricks for sugars? I'm not aware that there is an in-field measurement, although I do believe there are instruments being developed that will be available in a few years capable of doing that. But at this point, the only measurement that I'm aware of is collecting a leaf sample and sending it to a laboratory for a dry matter-based analysis, which is fairly commonly done, particularly on forages. Question from John Wilson. Hi, John. What are the main strategies to increase lipid levels? John, it's really simple. We need two things. One is we need very strong photosynthesis, and we need good biological activity in the soil. When we have those two, then lipid levels will go up all on their own. Question from Andy Harper. How do you measure a plant's photosynthesis? So there are instruments that you can measure it in the field on a research basis. They're fairly expensive, and I know the name of them, but I'm forgetting it right now. I don't have it off the top of my head. An analog that you could use, one fairly crude analog would be to measure Brix levels, although that's not something that I'm a particularly big fan or advocate of because of how variable and inconsistent they can be. The research instruments to measure photosynthesis in the field are fairly pricey. I want to say $50,000 to $60,000 range, and they actually measure the quantity of carbon dioxide absorbed, the quantity of oxygen released from a given area of leaf surface. So they measure the plant's actual photosynthetic ability and photosynthetic volume. So that's the best answer I have for that at the moment. Question from Michael Grove. How long does it take to get from seed germination to glossy, waxy sheen on the plant as a result of high photosynthesis? Michael, I would say that can happen immediately. The moment you have a plant, if you have really high seed quality is a factor here. If you have really high seed quality, then you can have that glossy, waxy sheen from the moment the seed germinates. If you don't have high seed quality and the seedling starts out needing to overcome that lack of support, it can still happen in as little as 2 to 4 days. So when you have good photosynthesis, that can happen very quickly. Question from Jim Dunlop. Hi, Jim. How does the plant obtain minerals if they are not naturally present in the soil, as in molybdenum and cobalt, and I might add iodine and selenium and others to that? That's a tough one, Jim. The answer is that they require these trace minerals only in very tiny concentrations, usually a few parts per million or perhaps even part per billion concentrations, but they are essential, they are required. And if they're not naturally present in the soil, then I believe this is something that we need to add. And there does seem to be evidence that there is enough in the soil that is locked up in the mineral reserves, the mineral matrix, at least in most soils, that as we develop really good biological activity, they can start releasing them. But that often takes time. It might take a few weeks or a few months, depending on the soil that we're talking about. So in that interim period, I think it's important to add those trace minerals. And you can add them using broad base materials such as seaweed or ocean products or humic substances that might contain them. But in addition to that, particularly when we're growing a high value crop, I think it's necessary to test those, which we do on a sap analysis. And when they're not present, we need to add them. It's a very good question. Question from Dennis Demmel. Hi, Dennis. How important is breaking hardpan to improve photosynthesis with gas exchange? It's foundational, Dennis. It's absolutely foundational. I forget, I think it was in a recent interview with Tineo and perhaps also in the last webinar. I forget where I recently was speaking about compaction, but we need to have very good gas exchange from the soil and the ability to release carbon dioxide. So if we have a hard pan, and of course we don't have good gas exchange, I personally believe that we cannot have healthy crops and we cannot have a truly sustainable ecosystem until we get rid of the hard pan. And that doesn't matter whether it's 6 inches deep or 12 or 4. The shallower it is, the bigger of an impact it has, but it's still imperative to remove it. Even in no-till systems, we need to remove it, and I believe that we need to remove it with iron. Remove it once, get rid of it, and then use plant roots and cover crop roots to make sure that they're not reformed. But it's simply removing them with crop roots and cover crop roots, in my experience and my observation, takes too long and costs too much. So we get a much bigger economic response, the crop response, if we remove it upfront using equipment and machinery. Question from Olivier: what kind of plants with enough lipids to be used in cover crops? So the first part of that answer is that any plant can produce high lipid levels. Grasses, legumes, alfalfa, clovers, etc., buckwheat. The lipid concentrations in any plant are simply a reflection of the overall plant health. With that being said, there are some plants that we know of that naturally have higher lipid levels inherently. Examples would be brassicas, such as kale and cabbage, cauliflower, etc. That waxy, oily leaf surface is an expression of a higher lipid content. Question from Greg Pennyroyal. Hi, Greg. With the difference in the formation of fulvic acid versus more stable humic substances, should we manage high temperature soils differently than more temperate soils? Well, not necessarily. I would have to think about that for a little bit and to come up with a more thoughtful answer. But I was immediately going to say that, well, first of all, we should, in higher temperature soils, we should keep those soils covered with plant residue all the time to keep them cool. And I believe the same to be true in a more temperate environment where we have lower winter temperatures as well. Those soils should also be covered. I think keeping those soils covered becomes more important, and also understanding that our objectives should be different. On more tropical soils that are exposed to higher temperatures, we shouldn't necessarily have the same goals of having 9% or 10% organic matter or higher. Our goals instead should become to have very high levels of microbially active carbon, extremely active levels of bacteria and fungi in the soil, because we can have an even more active microbial population than we can have on the soils with colder temperatures. So just understanding that the ecosystem is different, should be different, and should be managed slightly differently, or that, not that it should be managed differently, but that the goals should be different and should not likely be the same. Question from Riley: when transitioning a broadacre corn bean field from conventional management to using AEA products such as the Fall Soil Primer and BioCoat Gold, what are realistic first-year expectations? It's commonly mentioned that biologically active soil require fewer or no outside fertilizer inputs. How can a farmer know when he or she is able to begin cutting back on fertilizer applications? Riley, this is an awesome question. It's very challenging to make general statements about possible first-year expectations because of the starting place that we're beginning from. As you can imagine, many soils are in very different conditions from the first initial point, and so therefore the expectations for the outcome in the first year should be different as well. The way that we've managed this is, first of all, we do not suggest that we go to zero fertilizer inputs immediately. We have to earn the right to do that by building biology to be able to supply whatever is not coming from a bag or a tank. So the way that we manage it, if it's at all possible, we begin by putting on initial applications at the beginning of the season that are needed to get the crop going for the first, let's say for the first 60 days. And then we evaluate the crop response using sap analysis and perhaps using Haney analysis in the soil to be able to calibrate what the soil's capacity for nutrient release is for the remainder of the season. And then we side dress as needed, either with overhead irrigation where that is possible. If that's not possible, we will make recommendations for side dress for 1 or 2 applications. And usually what we find is that when we measure the actual crop requirements as measured on a sap analysis, and we apply only what the crop requires that the soil isn't capable of delivering, we're often only putting on sometimes as little as 50%, sometimes as much as 70% of the historical nutrient applications in prior years. This is particularly true for nitrogen. So we evaluate the results and the success in the season using SAP analysis so that we don't guess to say, oh, we think this year you can use 30% less and then end up being in trouble because of it. It's much better to have the tools to be able to backfill if needed in season and to be able to measure exactly where the plants are. I think that's a very important part of the success that we've had is not guessing. Question from Mr. Anonymous. I farm in a tropical region where it's very difficult to build organic matter. Temperatures are high and most organic matter degrades quite rapidly, leaving little organic matter to cover the soil. In the dry season, it gets very dry. I am worried that applying Spectrum and Rejuvenate will kill the beneficial microbes with high temperatures, dryness, and sun. I'm considering using covering with wood chips or introducing organic matter that will take longer to degrade. So wood chips with Spectrum and Rejuvenate. What are your thoughts on this? I think adding more complex carbons, such as wood chips, can be a great idea. And I'll also mention that dryness doesn't kill microbes. It makes them go dormant, but dryness in and of itself, dry soils in and of themselves don't kill biology. What kills biology is high temperatures. So if you have very warm soils and that are exposed, then that certainly lead to reducing soil microbial populations. Question from Kevin Tolley. Hi, Kevin. If you were to do one thing on a cover crop in the fall, would you apply a foliar or a soil primer? I would imagine a foliar based on this presentation, but in late fall, would you shift towards a soil primer once the crops are slowing down? So, Kevin, it's a great question. I would generally, if I had to choose between the two, I would select to do a fall primer. And that is partially because generally in the fall, cover crops are still relatively small, and putting on a foliar application isn't going to have nearly the response that it might when the crop is larger and has more biomass in the spring, for example, or in the early part of the year. So for a fall application, I would generally prefer to put on a fall primer. Very good question. Another question, if cover crops are in use, would increasing photosynthesis in the cover crop and cash crop be beneficial, or only increase cash crop photosynthesis and not use cover crops? So first of all, I'm not suggesting to not use cover crops. I think they're a very important tool that we should use as much as possible. And the ideal, the ultimate ideal, which is economically justifiable in high value crops, is to work on increasing photosynthesis in both your cash crop and in your cover crops. So we actually have a number of fruit and vegetable growers we work with that foliar feed their cover crops because they want the high lipid content and to be able to build soil organic matter quickly. But the answer is any green plant that is growing, you can put on foliar applications and increase the photosynthesis of those crops and get an organic matter response. Going through some of these other questions here, what is a good way to improve CO2? I realize I didn't answer that specifically. It could be a whole conversation in and of itself, but very briefly, carbon dioxide is supposed to be delivered from microbial respiration, from bacteria and fungi in the soil, consuming root exudates and other organic matter, and releasing carbon dioxide during the growing season. That's the best way to improve CO2 is to have really active biology. Question from Andrew Johnson. Hi, Andrew. If soil is adequate in manganese and iron, but plants are deficient, how do we facilitate uptake by the plant? The reason many soils are high in manganese and iron, but plants are deficient, is because the manganese and iron that is present in the soil is primarily in the oxidized form, which is a form that is not physiologically active within plants. So again, it is microbes, particularly bacteria, that are responsible for converting the oxidized minerals to the reduced form that plants can absorb. So again, it really comes down to soil biology. You have really aggressive, vigorous biology, and you'll get really good absorption of manganese and iron. In fact, I think in the podcast interview with Gary Zimmer, or perhaps in a private conversation, might have been in a personal conversation, Gary described how they actually use manganese levels on a tissue analysis, on a forage analysis for dairies. They use manganese levels on a forage analysis as an analog for microbial activity in the soil. When you have really high microbial activity, manganese levels go up. When you have poor microbial activity, manganese levels will stay down. Question from Barbara. What does the crop's ET tell us about plant health? Barbara, this is a fascinating question. I haven't thought of it quite in that way before, although we've certainly talked about water use in the past. So ET, evapotranspiration rate, we know that when we have healthier plants, they have a proper balance of nutrients, they're forming higher lipid levels, they will respire significantly less, and they require significantly less water, as much as 50% less, or as much as only 50% of what we consider there to be their common requirements. So I would suggest that the evapotranspiration rate, the lower that ET rate is, the healthier the plants are. And of course, more growers are beginning to measure ET, but most commonly, most growers don't actually measure ET. They simply use an estimate or a standard number, and that isn't consistent across the board. Question from Lawrence. Can the building of soil organic matter occur in very sandy soils with low biology as quickly as you mentioned? So, Lawrence, this is a good question, and I've mentioned it in the past in prior webinars. We've covered this briefly as well. Let's just say we should have a different goal in sandy soils. The goal is not to have very high organic matter, 9 to 10%, or even 5 to 6%, but to have very high levels of microbially active carbon. So our total organic matter number is likely to be lower, but our microbially active carbon, as measured on a Haney analysis, can be much higher. So we should have a different goal and a different way of measuring success in very sandy soils. Question, if for optimal photosynthesis, the right amount of minerals is required, doesn't that indicate that we need good soil first in order to grow healthy plants? Good question. Well, the conundrum contained within your question is that the release of minerals that are in the soil profile requires the presence of biology, bacteria and fungi, to release those minerals. And bacteria and fungi require the presence of a food source, which is plants. So when you go back, you are correct in that it is a synergistic system. They are both designed to work together and for both to complement each other. The point that I've been seeking to make is that the engine which drives the system is photosynthesis. It's plants, not biology. Question on fall fertilizing. Can organic fertilizers be applied in the fall for nutrients to be available in the spring? I would say generally yes. And generally, when you have, I do not believe that nitrogen applications in the fall, particularly non-organic nitrogen forms, is a good idea at all. But phosphorus, potassium, etc., Phosphorus can become locked up in reserves, but potassium and other nutrients, when again, predicated on good biology. When you have good biology, fall applications of nutrients should remain very available for the following growing season. I mentioned the role of iron in reducing a hardpan. Could I elaborate? I was referring to iron in the hard physical form of steel, using machinery, using equipment to break up that compaction layer for the very first time. I have a few questions here that are off topic. Let me see if there's any more that are on topic. Question on winter rye. I have winter rye planted now and applied the fall primer. Should I use accelerate, put on a foliar application of accelerate on the spring before plowing it for corn? There's too many things I don't know about the rest of your operation to be able to answer this question well, Murray, such as how long it'll be growing in the spring and at what stage you plan to incorporate it, how far north or south you might be. But if you are capable of getting 18 inches or more of growth before you incorporate it, then you are likely to get a lot of value in building soil organic matter from putting on a foliar application next spring. Now, you mentioned that it's for before planting corn, and if you're growing commodity conventional corn, then we would need to look at economics very closely, and there's probably a better place to get an immediate economic return than that foliar. You'll get a long-term soil organic matter return, but you might not get as an immediate economic return. Corn, as we know, is very tight margins. If you're growing organic corn or if it's food grade corn, then that changes the equation completely. But I would probably be cautious about putting on that application on commodity corn. If you're growing a high value crop, that changes completely.
John, I have a question here from one of our phone call-in listeners who asks, if calcium is deficient, how does that change hormone function and sugar movement to roots versus shoots?
Okay, that's a good question. So I think the rest of the questions here are off topic of what the webinar is about, but we still have a number of them. There's 8 of them here, and Anna just asked another. So we'll be bouncing around a little bit, but I'll go through and answer the rest of these questions as well. So to answer the calcium and hormonal question, I answered this question in some detail in the podcast episode on vegetative versus reproductive nutrients. The short answer is that calcium will drive a large amount of vegetative biomass, but it also has a strong synergistic relationship with the hormone cytokinin. So calcium actually drives cytokinins, which drives strong reproduction and strong root growth. So calcium is the foundational element needed if you want to build a lot of biomass, but not lose reproduction. Hope that answers the question well. And you can refer to the podcast if you want more details or more elaboration on that. So there's another question here. What practices, nutrients, and inoculants can we use to have a smooth transition transplanting crops from greenhouse to the field? It seems having robust seedlings helps set up plant life and helping root establishment. So we have a transplant program that we put together that I would suggest you look at and refer to. But in brief, I believe that it's important to inoculate transplants with both bacterial and fungal inoculants, mycorrhizal fungi, etc., even when they are present in the greenhouse media, because the peat moss-based media or even compost-based media support a different microbial profile than the real soil. And so I believe that it's important, even if they were inoculated in the greenhouse, to re-inoculate them again at transplanting. Then, of course, using seaweed is an old standard standby, and not using water-soluble phosphorus fertilizers, and using ocean minerals, humic substances, and calcium. And then there can be more that varies based on the specific type of crop. And just to expand on that a little bit, one of the biggest challenges with seedlings is the way we think about growing seedlings has become really screwed up. Seedlings in the greenhouse should be grown with a nutrient ratio, a nutritional profile of very high calcium, low nitrogen, low potassium, and high phosphorus. So high calcium, high phosphorus, low nitrogen, low potassium. When you do that, you will have very short, stocky, sturdy seedlings with very large stems that do not have to be hardened off. They can be taken straight from the greenhouse and transplanted to the field with no transplant shock because they don't have weak watery cells like a seedling that has a high nitrogen and potassium content will. Another question. I'd like your opinion on primary tillage for an organic dairy farm. The grass we plant ends up becoming our biggest weed in our corn crop. This is a big question and something that I think would be worth having an offline conversation about. I would suggest you find ways to suppress the growth of your grass crop outside of tillage, which is very possible. There are tools available today, even organically certifiable tools, that you can spray onto grasses before you till that would largely shut down their growth or slow it down enough that they would only start growing late in the season to the point where they would not be competing with the growing corn crop for moisture and for nutrients. So that's the first thought that comes to mind. And again, I don't know a lot about the specific situation, have a lot more questions. But I would suggest that you may be positive. There are probably ways to figure this out without using a mold board. And I'll offer one more tidbit, which is that the single most profitable dairy farm that I know of grows no corn. Not just that dairy farm, but I would say the most profitable dairy farms in general that I know of grow no corn. So that can be something to think about also. Another question, can I raise our blueberry iron enough by a foliar and fertigation with rebound iron when we irrigate with very hard water, 294 parts per million? The answer is yes. It'll require slightly higher applications with very hard water, but the answer is yes, you can do it. Reverse osmosis water has been suggested, but is out of our price range at the present. Is our water completely confounding our iron amendment attempts? It'll slow it down, but it's still possible to put on enough iron to make sure the plant has a general supply. So it'll require slightly higher applications, but it's still very possible. Question from John Wilson. Would you advise feeding the plants with foliar sprays, fermentations, in order to have a healthier plant and therefore contribute to healthier soil? Is that the main benefit of these fermented biofertilizers? John, the answer is yes. Foliar applications of nutrients and trace minerals are, I believe, one of the keys tools to increasing photosynthesis and to hacking the system. Question, do we have a recommendation for a small-scale reverse osmosis filtration system? Yes, I would recommend contacting Persanova, which you can find online, P-U-R-S-A-N-O-V-A. They now have smaller-scale units that are very affordable. How does photosynthesis efficiency affect garlic bulb sizing? Is there an optimal time to apply foliars of the photosynthesis nutrients? So obviously, the quantity of photosynthesis has a direct correlation to bulb sizing. The more sugars you can produce during the bulb fill stage, the larger the bulbs are going to be. And the optimal time to apply it is when the crop needs it, and that varies. There's no real way of me knowing that. In general, the greatest need occurs during the fruit fill or grain fill or bulb fill stage for whatever crop we're talking about. And that's when we have the greatest economic impact, is during the actual fruit fill stage. Question from Tom. When do you recommend the first foliar application of trace minerals on broadacre crops to begin maximizing photosynthesis for corn and soybeans specifically? For corn, we get the biggest responses between V4 and V5. We've done it as late as V6. And for soybeans, the first trifoliate is when we get the biggest yield response. Good question, Tom. Thank you. Follow-up question from Tom. How many acres should one sap analysis represent, and which lab do you recommend? The only lab that we use is Crop Health Labs in Belleville, Ohio, which is a purveyor or marketer for Nova Crop Control in the Netherlands. And how many acres? We've done as little as a few hundred feet or one greenhouse, and as much as 640 acres. So it's one management unit. Whatever you are managing as a unit can be represented by one SAP analysis. Follow-up question from Riley on SAP analysis. You have several times mentioned using a sap analysis to determine what nutrients a plant needs. What methodology was used to determine the ideal sap nutrient levels for each crop? Riley, this is an awesome question, a question I wish more people would ask. The desired values for the nutrients, we've calibrated all these desired levels for all the crops that we've worked with here in the U.S., which is I think about between 40 and 50 crops, almost 50 crops at this point. And those nutrient levels are not based on laboratory averages. They're not based on field averages of the averages of all the hundreds or thousands of samples that we've taken of a given crop at a given growth stage. Instead, they are framed around what are the best fields, the highest yielding and the highest quality fields. The fields that have the greatest disease resistance and insect resistance and that produce the highest yields. What did the nutritional profiles look like there? So we have enough of those that we have strong database of these really healthy fields to support what a really healthy plant should actually look like. So the nutrient levels are not based on averages. The desired values are not based on averages. They're based on what we believe the optimum to be. And one last question, is there a direct correlation of plant health and bionutrient levels, organic versus conventional vegetable production? So Phil, from my perspective, those are two different questions. There is no correlation between plant health and organic versus conventional. There's no correlation between bionutrient levels and organic versus conventional. So to your first question, is there a direct correlation of plant health and bionutrient levels? I believe the answer to be yes. Many other people believe the answer to be yes, but there is still a big gap in documenting and quantifying all the ways for which that is true, because we're talking about hundreds, or in many cases, thousands of different bionutrient compounds that are different for each species, or I should say occur in different concentrations for each species, for each variety, even in some cases. So I believe, yes, there is a direct correlation, and we have a fairly accurate meter, a qualitative meter, in evaluating bionutrient levels that is simply called the nose and tongue. So differences in flavor and aroma can definitely be attributed to plant health and to nutritional profiles. And now we simply need to quantify exactly to what degree these various bionutrient levels are varying. And there is no correlation between plant health and bionutrient levels in organic versus conventional. Some of the highest quality, most aromatic and flavorful fruit that I've seen has been produced conventionally. Some of it is also being produced organically, and then there is also very poor quality organic and very poor quality conventional vegetable production. All right, so those are all the questions that have come through. I want to thank all of you for participating, for asking questions. I hope that you found it useful and enjoyable. Thank you very much for participating, and we will see you next time. Have an awesome day. Happy growing. Thanks, everyone.